171. Which is the most important factor for the continuity of a species from an evolutionary perspective? What is more important for the continuity of a species? Replication of genetic material, formation of gametes, synthesis of proteins, none of these. Continuity of a species in terms of evolution is the replication of genetic material. The genetic material should replicate, and that will help the species remain a species. Without DNA replication, you cannot maintain the DNA content and the chromosome number. Random genetic drift in a population most likely results from. Why would the genetic drift happen? Highly genetically variable individuals, interbreeding within the population, constant low mutation rates, small population size. What can lead to random genetic drift among the four? See, what will not lead to genetic drift? Equilibrium will be maintained under which conditions? Opposite of that will lead to easy genetic drift. So, which of the given four conditions is more likely to lead to genetic drift? Change of the genetic pool. High genetic variability is more likely to keep the balance constant. Interbreeding within the population. There is no selective breeding, but within the population, if there is interbreeding, there is no exit of the genes or no movement of the genes outside. Low mutation is also equilibrium. Small population size is more likely to lead to genetic drift. Why? Why does a small population favor genetic drift, not equilibrium? See, in any population there will be movement. In any population there will be movement. When the population is small, movement of individuals or elimination of individuals from the population can significantly affect the gene pool, because the number of alleles available in the gene itself is small. Alright, next. What if a set of individuals move out of a population and become the originators of the genetic pool in a different location? What do you call that? Founder effect. Founder effect. What is it called when a large part of the genetic pool is eliminated, either because of some disaster or mass death or disasters, and only a small set of genes are present in the population? Bottleneck effect. What is bottleneck? There is a wide pool, but what is available in the population is a narrow band of genes. Why? Out of that wide pool, there might be elimination of population, disasters and mass removal of populations. So you have to remember founder effect, bottleneck effect, genetic drift. Which of the following does not support Lamarck's theory of inheritance of acquired characters? See, Lamarck's theory is accumulated variation, correct? Accumulated variation. So variations accumulated over a period of time will become constant. And Lamarck's theory is based on utility. Changes which are useful for the population will remain. Changes which are harmful for the population or organism will be eliminated. Now let us see the examples which does not support Lamarck's theory. Lack of pigment in cave-dwelling animals. Can it be explained by Lamarck's theory? Cave-dwelling animals do not require pigments. Darkness. So pigments will be removed. Melanization in peppered moth. Can that be explained by Lamarck's theory? Can that be explained by Lamarck's theory? Melanization in peppered moth. Lamarck's theory is based on use and disuse. what is the moth using so that it is developing the pigmentation? It is not using anything. Alright, so Lamarck's theory cannot explain melanization in peppered moth. Absence of limbs in snake can be explained because snakes move in the cracks and crevices. They always crawl. And because of that, there is no utility for the legs. Disuse of legs leads to elimination of legs. Presence of webbed toes in aquatic birds. Constant stretching out of the toes led to the development of webs. All of that is explained by Lamarck's theory. Melanization in peppered moth cannot be explained by Lamarck's theory. Lamarck is use and disuse. The biological concept of species is mainly based on biologically, species is something which has reproductive isolation. Is reproductive isolation unbreakable? Is reproductive isolation unbreakable? Meaning, can there be interspecific breeding? Can there be interspecific breeding leading to viable offspring? Firstly, reproductive isolation is at multiple levels. One, gamete mismatch, organ mismatch, then survivability of the embryo, fertilization barriers, and even if the fertilization happens, survivability of the embryo. Even if the embryo survives, reproductive ability of the offspring. At multiple levels, there is a protection, there is reproductive isolation for the species. Interspecific hybridization in animals might not lead to viability, reproductive viability. So in that sense, even if there is a offspring because of interspecific cross, it may not be sexually or reproductively viable. So reproductive isolation is the biological concept, is the basis on which the species is defined. Short-lived immunity acquired from the mother to the fetus across the placenta or through mother's milk to the infant is categorized as innate non-specific, active immunity, passive immunity, cellular immunity. Passive. Is it specific or non-specific? Passive immunity is specific immunity or non-specific immunity? Passive immunity is specific or non-specific? See here, there are two ways of looking at things. One, immunity specific, non-specific. Specific is always mediated by lymphocytes. Non-specific is neutrophils, macrophages. So specific immunity is the attack is dependent on who is being attacked. It is dependent on the invader. And every new invasion of the same invader will lead to increased intensity of attack. So the first time enemy comes in, you identify the enemy, you beat the enemy and you remove the enemy. The next time enemy comes in, your attack will be even more stronger. With every subsequent attack by the invading organism, the immune system will respond more aggressively. Here it doesn't, in non-specific case, it doesn't matter. Who is coming is not a factor here. It is a foreign body is all that matters. If somebody who is foreign enters, these fellows will attack the same way. They are cold-blooded murderers. Non-specific immunity is cold-blooded murder. Doesn't matter who is being killed. You don't belong here, we will kill you. That is non-specific. Specific is, how many times should I tell you? Range. Did I tell you earlier not to come? Why have you come? Next time he comes, you get more angry. Every time the foreign body is coming, you are getting more angry. Here, again, lymphocytes, direct attack, antibody mediated. Direct is cell mediated. So this is T lymphocytes, this is B lymphocytes. T lymphocytes and B lymphocytes. Alright? Here again in specific you have active and passive. Active and passive is specific only. It is of lymphocytes. When these fellows are produced and activated and they mount the attack, it is active. When you directly give antibodies, to tackle the antigen, it is passive. Understand? Active and passive both are specific. Active and passive both are specific. Non-specific is only neutrophil macrophages. Is that clear? So, if the options were, see, innate immunity is non-specific only. Innate immunity is non-specific. Specific immunity is acquired. Specific or acquired, non-specific or innate. Non-specific or innate. Now, innate non-specific immunity is option 1. Active immunity is option 2. Passive specific immunity, if it was option 3, and option 4 was passive non-specific immunity, then it would have led to more confusion if you did not have clarity here. Probably many of you would have chosen passive non-specific. Passive and active both are specific. In active immunity, the cells of the body will produce the antibodies. The lymphocytes, B lymphocytes within the body produce the antibodies. In passive immunity, ready-made antibodies are given from outside as injections. You give ready-made antibodies. So you are not wasting time in the body trying to identify the antigen, develop the response, produce antibodies and tackle the antigen. You are directly giving antibody. When do you want to do this as a treatment? When do you want to do this as a treatment? When the infection load is high, when the infection status is not known and you don't want to take the risk of the window of stimulation of active immunity. Let's say like rabies. If a rabid dog bites and you don't know what the status of the dog is, it's a, let's say it's a stray dog, it's a street dog, so it bites the patient. It has bitten closer to the head and neck. Let's say it has bitten the shoulder or the neck or the face. We don't know the status of the dog. Vaccination has to be given to stimulate the immunity against rabies. But vaccination will take 5 to 7 days to become effective, to start producing antibodies. If at all rabies virus was present in the dog, if the dog was rabid, closer the bite is to the brain, higher is the risk. So now the dog has bitten the shoulder or the face. We don't know the status of the dog. You don't want to take any chance. So what do? You will give anti-rabies antibody. Anti-rabies antibody. We are assuming that the dog is rabid. We give antibodies so that if at all the virus has entered the body, we are already giving antibodies so that the antibody will remove the antigen. Plus vaccination is also given so that backup immunity is free. Understand? There is one physiological condition where antibodies are given. Which one? To prevent, as a preventive measure, antibody is given. Erythroblastosis fetalis. Same case, no? The mother is Rh negative, the fetus is Rh positive. During the end stages of pregnancy, fetal blood enters the mother's body. If the mother's immune system identifies it, the mother will develop the ability to produce antibodies against Rh antigen. We don't want the mother's body to identify the Rh antigen. So you give antibodies externally so that Rh antigen is removed. Now protecting the next baby from an attack by the mother's immune system. Okay. The first baby will be delivered, the baby will be safe and sound, but the second baby will get attacked because once the baby's blood enters the mother's blood, if at all the mother is already sensitized to the Rh antigen, quickly antibodies will be produced and employed, and IgG antibodies are produced. IgG can cross placenta. IgG can cross placenta and enter the fetal circulation, attacking the fetal RBCs. That should not happen. So by the time the delivery is initiated and the delivery happens, sufficient antibodies can enter the blood of the baby, causing significant damage. To prevent that, we give antibodies. Passive immunity. Be very clear about this. This is a simple concept, but confusion is possible. So in this case, across the placenta, what kind of antibodies pass? What kind of antibodies cross the placenta? What type? What type of antibodies cross the placenta? Name them. IgG. What type of antibodies are given via breast milk? IgA. Which antibody is surface antibody, which acts on the surface, mucosal surface? IgA. IgA is surface antibody. See, I have told you this. Why breast milk contains IgA? It goes into the gut of the baby. Baby is more likely to put its hand in the mouth. We don't know what infection can go in. So protecting the surface of the baby's intestine by the IgA antibodies of the mother, so that the baby does not develop diarrheal disease. Diarrheal disease in the infant stage, in the early life of the baby, can be fatal. The first year of life is infancy. During infancy, diarrheal disease can be fatal. 2-3 episodes of diarrhea can put the baby into dehydration. So see how nature has protected. Nature has created these mechanisms to protect the baby. Next. HIV has a protein coated genetic material, which is genetic material of HIV is single-stranded RNA plus a special enzyme. What is the enzyme? Special enzyme in HIV? Reverse transcriptase. In the practice of mating animals within the same breed but having no common ancestor, same breed, no common ancestor, in this side of the pedigree up to 4 to 6 generations is outcross, crossbreed, inbreed, interspecific hybridization. What is it? Outcross, crossbreeding, outcrossing, crossbreeding, inbreeding, interspecific hybridization. See, what are the possibilities of crossing an organism? Same breed, related. Same breed, unrelated. Different breed. Different breed, same species. Different species. That's all, no? These are the possibilities. These are the possibilities with, you can cross one organism with another organism. Same breed, related. They have common ancestry within the 4 to 6 generations. Unrelated is same breed with in the previous 4 to 6 generations, no common ancestry. Different breed of the same species. Different species. What is this? Inbreeding. Same breed, unrelated. Outcross. Different breed of the same species. Crossbreeding. And different species. Interspecific. Hybridization. That's all. Okay. Now what is this? Same breed but having no common ancestors. Outcross. Outcross. These are the possibilities. Write down. Telling you, you know that I am telling you, you are just not lifting up the
Yes, I cannot see, sir.
You cannot see? You heard it from same breed, related species? Oh, you are zooming in and seeing it. Okay, next. The World Biodiversity Day is celebrated annually on 29th December. I have made note of it. Fifth June, I know. What is 5th June? World Environment Day. 21st June. International Day of Yoga. That's all. Those 2 dates I know. On 79. Animals take phosphorus from? Animals take phosphorus from? The source of phosphorus for animals is plants only. Why do we need phosphorus? Why do we need phosphorus? Bones, ATP, DNA, all of that is phosphorus. No? There is phosphorus in all of that. Biomagnification of DDT causes a decline in bird populations by disturbing calcium metabolism, thinning of eggshells, premature breaking of eggshells, all of this. DDT disrupts all of these processes. Okay. Done. 53 weeks. Bryopteris, Pteris, Adiantum, Lycopodium. Bryopteris, Pteris and Adiantum, all 3 are ferns. All 3 are ferns. Adiantum is called maidenhair fern. Bicollateral conjoined vascular bundles have bicollateral conjoined. There are two things. Collateral means what? Xylem and phloem are on the same axis. That is collateral. Bicollateral means something is in the middle and one is on the either side of it. Conjoint means they are close, they are attached to each other. Let us see. Bicollateral conjoint vascular bundles have xylem and phloem which are arranged in an alternate manner on different radii. Different radius means it is radial. So A is not right. The xylem and phloem which are situated at on the same radius, and it has two groups of phloem along the two sides of xylem, inside and outside of xylem. That is correct. Xylem and phloem in the same radius, but it has only one group phloem outside the xylem. That is collateral. That is not bicollateral. Phloem surrounds the xylem tissue. That is, what do you call it when phloem surrounds the xylem completely? One is at the center, another one surrounds it. So what kind of a vascular bundle is that? Concentric amphivasal. Okay. If xylem surrounds the phloem, it is also concentric amphicribral. Alright. That's all. Then with vascular bundles, you also have to know closed and open vascular bundles. Closed vascular bundles do not have cambium in between xylem and phloem, so there is no secondary growth. Open vascular bundles have cambium in between xylem and phloem. Which of the following processes make direct use of oxygen? This, you are all masters of this. Glycolysis, fermentation, electron transport, Krebs citric acid cycle. Which of these use oxygen directly? Electron transport chain. Electron transport chain. Do we need oxygen in Krebs cycle also? Do we need oxygen in Krebs cycle? Don't need oxygen in Krebs cycle directly. See, but oxygen is removed in Krebs cycle, no? Carbon is removed as carbon dioxide. Carbon is removed as carbon dioxide. Oxygen is required for Krebs cycle, but not as direct molecular oxygen. The method of inducing early flowering by pre-treatment of seeds with a certain low temperature treatment of seeds is referred to as vernalization. Photoperiodism is photoperiodism determines what? The nature of plants in terms of light exposure. Long day, short day, day neutral plants. What is abscission? Breakdown. Breakdown is abscission. Phototaxis is plant movement in response to light. The technique that was employed to produce haploids of Datura was, by what do you produce haploid embryos among the given four options? By anther culture. Meristem culture will give rise to what? What will you get if you do meristem culture? Virus-free plants. Virus-free plants. If you do embryo culture, why do you want to do embryo culture? Embryo culture is done for rescuing the embryos. Vulnerable species, rescue the embryo, do embryo culture. Anther culture is for haploids. Protoplast culture is for somatic hybridization. Protoplast culture is for somatic hybridization. SCID is caused by defective gene coding for the enzyme called, which enzyme? Adenosine deaminase. SCID is severe combined immunodeficiency. The leaves are modified into tendrils, hooks, pitchers and bladders in the following plants respectively. Yesterday in the previous paper we saw tendril modification in sweet pea. So sweet pea, bladder, bladder and picture. Nepenthes is pitcher plant. Nepenthes is pitcher plant, no? So, tendril is sweet pea, picture is Nepenthes. Only one option we have. Sweet pea, cat snail, Nepenthes, urticularia. Both pitcher plant, Nepenthes and urticularia are parasitic plant, not parasitic plants. They are predatory plants. Predatory plants. Why do plants develop the predatory habit? What is the need for plants to develop predatory habit? For what? Why is this pitcher plant or bladder plant eating insects? They prepare their own food, no? Then why do they have to capture food? Animals, greedy plants. Why? What is the use? If it is useful, then why are the other plants not doing it? What is the purpose of catching insects? For pollination. You want insects to carry the pollen. You eat away insect, what pollination? Why do you think? These plants are eating insects for fulfilling a nutrient deficiency. Where they have evolved, they don't get a particular essential nutrient. To fulfill that need, they have developed the adaptation to capture animals, capture insects, and dissolve them, digest them, and get that nutrient. What nutrient? What nutrient? What macronutrient? Now you cannot say out of syllabus. Today's paper, there were no out of syllabus questions. Finally, I am saved. Okay. Hopefully, the remaining papers will not have out of syllabus paper questions. So, out of syllabus questions creeping into the question paper has become the source of joke for all of you. That I realized yesterday. Suddenly something was understood by everybody and I did not understand that. It means that has been the point of discussion. Already. Why? What nutrient? Nitrogen. Nitrogen. To fulfill the need for nitrogen, to fulfill their nitrogen requirement, they do predation. There are 4 macronutrients: carbon, hydrogen, oxygen, nitrogen. Correct? Those are the 4 major macronutrients. Carbon, hydrogen, oxygen, you cannot have deficiency. Hydrogen and oxygen is available in water and carbon is available in the form of carbon dioxide. Nitrogen is the one which is very crucial for plants. So nitrogen becomes the limiting factor for their growth, and where these plants have evolved, they don't get nitrogen in good supply, so they develop this adaptation to eat these insects. A lysogenic cycle involves, lysogenic cycle is found where? Who has lysogenic cycle? Viruses. Viruses have 2 types of life cycle: lytic life cycle and lysogenic life cycle. Lytic life cycle is, see, ultimately what is the virus, what does the virus want? It wants to get into a host, use the machinery of the host, multiply its genetic material, protein coat, assemble, burst open the host, come out, repeat. So virus is getting into a host to eventually break the host and come out and infect more hosts. When the virus is breaking the host, it is called lytic life cycle. Lysis means to break down. It is possible that the viral genetic material continues to remain within the host and multiply along with the host without breaking the host open. That is called lysogenic cycle. When the virus is not rupturing the host to come out, it is going along with the host and multiplying when the host multiplies. Does that mean the virus will continue to do it forever? It is only a matter of time before the virus starts utilizing the host and breaking it. Okay. So eventually a lysogenic cycle will become lytic cycle. There is no escaping that. Embryo sac diagram. Identify A, B, C, D, E, F. A is synergids. B, polar nuclei. C, central cell. D, antipodals. E, belliform apparatus. F, egg. So no confusion there. Embryo sac is very well understood. The genetic alteration of a cell's genome is called, genetic alteration of a cell's genome is called what? Transformation, reverse transcription, genome conversion, invasion. It is easy. Reverse transcription is RNA to DNA. Reverse dogma, molecular, what is that? A central dogma reverse. Reverse transcriptase will do central dogma reverse, RNA to DNA. Otherwise, the flow of information is from DNA to RNA to proteins. Transformation is seen in which organism? Genetic alteration by way of transformation is noticed in which organisms? Bacteria. Transformation is always beneficial, true or false? Transformation is always beneficial, true or false? False. It can be beneficial or harmful based on what the genetic material is. Conjugation, transformation, and what is the third one? Transduction. Conjugation is one bacteria has an advantage in the form of a plasmid. It will multiply the plasmid and transfer it personally to another bacteria. That is conjugation. Transformation is genetic material is present around the bacteria in the medium. It will pick it up and integrate it into the cell. So picking up stray DNA from the surroundings, that is transformation. Transduction is bacteriophage, this fellow doesn't want it, but bacteriophage will come, catch hold of it and inject it. That is transduction. Transduction. Learning from qualified teachers is conjugation. Verified knowledge is transferred, conjugation. Learning from friends is transformation. Whether what you learn is beneficial or harmful depends on what your friends are teaching you or what you are learning. Transduction is learning from antisocial elements. You don't go and learn. They will catch hold of you and they will give you. It is like starting to smoke or drink alcohol or getting into drug abuse under peer pressure. Doing something under peer pressure is transduction. Just because you want to belong to a particular group, you do something which is wrong. Transduction. Always harmful. So what do you have to do? When the situation of transduction comes, you should try to get away from the bacteriophage. Okay? You should identify there are bacteriophages. If you are a bacteria, there will be bacteriophages around you in the guise of a friend also. If a friend is like a bacteriophage, the option you have to exercise is not take what the friend gives you to not hurt your friend. Save yourself by getting away from that person who in the first place was not your friend. Alright? I've seen so many kids, just to be in a group makes them feel confident, A group makes them feel wanted, makes them feel important, attention is given. Just to be in the group, they try to do everything that the influential people of the group do. In any group, there will be one or two people who are the influencers. So you should be very careful. It's okay not to have a group, it's okay. Wow. What is this? I wanted to shoot away. Wanted to shoot away. It's just. Okay. Okay. Be careful. Be careful. Medical college is full of people like that. Not full of people like that. Full of opportunities like that. I should say. Not full of people like that. It's like some uneducated movie directors portraying medical college. Such pathetic portrayal of medical college. The way people dress in cinema, medical students. The kind of behavior that is shown of medical students in movies. Nothing like that. The most de-glamorized place for an institution is a medical college. No glamour at all. No flashy things at all. Hardly you will get time to tie your hair properly. Alright. You will be worse than unpaid slaves. Okay. So. Unlike the flow through vessels, water tracheids must flow through. See, tracheids are cells. Tracheids are cells with sclerenchyma. So there is no continuity of a passage for the water that flows through tracheids. But water flows from one trachea to another trachea in a zigzag manner, and it passes through what? What do you call the gaps in the tracheids? They are pits. Now tell me, pits are the places on the tracheids where there is absence of wall. True or false? Where the wall is absent, cell wall is absent. True or false? Pits are places where the cell wall is absent. Pits are the places where the secondary cell wall is absent. Lignification makes the cell impermeable. Nothing can pass if it is lignified. So pits are the place where secondary cell wall is not deposited. Primary cell wall still remains. Primary cell wall is permeable to water. It will allow water to pass. Gradient can be established along the primary cell wall. But secondary cell wall will not allow. So pits are the places where secondary cell wall is absent. Primary cell wall still continues to exist. Okay. Primary walls of the endodermis are impregnated with. What is the primary wall of the cell wall, endodermis loaded with? Suberin. Why? Suberin is a lipid. Suberin is a lipid. Suberin in the cell wall makes water not able to cross it. Endodermis, you should not have all the cells of the endodermis allow water to pass. Does that mean the entire endodermis has been made impermeable to water? There are certain cells in between which do not have suberin. What are those cells called? What are those cells called? Now if this is the epidermis, this is the steel, this is, let's say, endodermis. This is endodermis. The cells that I am marking, these are the cells which do not have suberin deposition. Why don't they have suberin deposition? And why the cells in between, all the rest of the cells here, the cells here, the cells here, all of them have suberin deposition. Only cells in between, some of the cells don't have suberin deposition. Why suberin deposition and why some of them don't have it? Root hair absorb water and minerals. All of that pass through the cortical cells of the cortex. Where should they reach? They should reach the vessels, xylem. They should reach the xylem. So they have to be transported. While they are passing through this part, they pass through these cells with using both the pathways. What pathways? Apoplast and symplast pathways. Along the cell wall and through the cell wall also. So all of this water is coming rushing through towards the steel. They are all coming rushing towards the steel. Adjacent to these cells which do not have suberin deposits are the xylem. Xylem is here. This is xylem. Now can you tell me why these cells don't have suberin deposit? So that they can allow water to pass. They can allow water to pass. Then why do these fellows have suberin deposits? So that water just doesn't flood into the steel, it is channelized towards the xylem. It is channelized towards the xylem. So water is coming from all around. Water is coming from all around. You want the water to go into the xylem. So what do? You put suberin deposits, suberin around the endodermis cells, except those cells which are adjacent to the xylem. So the water that comes here cannot cross this endodermis. What will happen? It will move like this and reach here, some of it will reach here. To channelize water into the xylem. That's clear? It is all logical. You don't have to memorize. Once you visualize it, you know it. Wax deposits are found in the cuticle. The cuticle has wax deposits, not the endodermis. Which of the following is not a true statement about the kingdom Protista? It is an artificial grouping. Yes or no? Protista is an artificial grouping. It is correct. Its members are placed there as a matter of convenience. Correct? Although diverse, the group represents a closely related group evolutionarily. True? Is it true? Although the organisms are diverse, they are all closely related evolutionarily. Is it right about Protista? They are not closely related evolutionarily. They are all distinct from each other evolutionarily. It contains unicellular, colonial and multicellular groups. Correct. In Protista you have variety of organisms. There is a Kannada movie, not a good movie, but a movie where the protagonist is an orphan. He doesn't have anybody, he doesn't have parents or siblings, anybody. He brings together different people of different age groups. He doesn't go around trying to get people to stay with him. But as he comes across people of different age groups in different situations, he brings all of them home. And that becomes a family. That's like kingdom Protista. They are not connected by any relationship to each other, but they are all conveniently grouped together. That is Protista. And all of these people, when they realize where they belong to or where they have come from or where they are supposed to go, they will be sent to the respective families, respective groups. That is Protista. They are all temporary location, refugee camp. It's like anyways, you understood. There is no need to create. Which of the following flowering arrangements promotes self-pollination? Monoecious, dioecious, dichogamous, none of these. Self-pollination is promoted in which of these? If a plant is monoecious, does it favor self-pollination or favors cross-pollination? Monoecious plants. If a plant is monoecious, what is favored? Self-pollination or cross-pollination? Self-pollination is favored. Monoecious means what? How many individuals do you need for representing both the sexes? Mono means one. That means what? Both male and female structures are present on the same plant. Dioecious plants are always cross-pollinated. Dioecious plants are always cross-pollinated. Monoecious plants always have bisexual flowers. True or false? Monoecious plants should have bisexual flowers. False. Not necessary. You can have male flowers and female flowers on the same plant. The necessity for a plant to be called monoecious is that both male and female reproductive structures are represented on the same plant. It is not necessary that all the flowers should be bisexual flowers. Both the sexes should be on the same plant, not necessarily on the same flower. Okay. What is dichogamous? Dichogamous. Dicho means two different. Gamous means gametes. So dichogamy is the property of one of the reproductive world developing earlier than the other. That means if androecium develops first, gynoecium gets delayed, so that the pollen cannot fertilize the gynoecium of this plant. So dichogamy is against self-pollination. Dichogamous condition prevents self-pollination. The usefulness of fermentation as a means of deriving energy is limited because it cannot generate enough ATP. It produces too much NH2. The end products are toxic to the producer. It uses more energy than it produces. What is right? The usefulness of fermentation as a means of deriving energy is limited. Why? See, the end products are toxic to the producer is not a necessary condition. Not necessary that everything which derives energy by fermentation is facing toxicity. Bacteria derive their energy by fermentation, lactic acid fermentation. What is the right choice here? It cannot generate enough ATP. Why? One molecule of glucose, when it goes through anaerobic fermentation, it gives only 2 ATPs. Whereas one molecule of glucose aerobically gives 38 ATPs. Okay? Diagram of a bacteriophage, in which one of the options are all the 4 parts correctly identified? A is, what is A? Head. B, sheath. C, collar. D, tail. So whoever has not received, please correct it now. Raise your hands if you have not received. Those, when madam is around, you please raise your hands and correct it. Don't wait for madam to come and check on your table. Do you have it? Have you kept it inside? Please show. Raise your hands, correct.